Sanger sequencing, also known as chain termination sequencing, is a method for determining the nucleotide sequence of DNA. Prior to performing this sequencing, DNA from samples needs to be isolated and purified. The process begins with mixing DNA template with primers, DNA polymerase, deoxynucleotides, and dideoxynucleotides. Primers are short, single-stranded DNA sequences designed to anneal to the specific region of interest on the template DNA. Primers provide a starting point for DNA synthesis. A DNA polymerase enzyme is also added to the mixture. It extends the primer, synthesizing a new DNA strand complementary to the template. Deoxynucleotide triphosphates or normal nucleotides, ATCG, used for extending the DNA strand. In addition to the normal deoxynucleotides, a mixture of dideoxynucleotides is added. Each DNTP is labeled with a different fluorescent dye, corresponding to one of the four nucleotides, A, T, C, G. DDNTPs lack a hydroxyl group at the 3' carbon, which causes termination of the DNA strand when incorporated, so the fluorescence is detected. This is the key to the chain termination mechanism. The mixture undergoes thermal cycling to perform DNA replication in multiple rounds, like the polymerase chain reaction. This step consists of three main phases. The first step is denaturation at high temperature. The double-stranded DNA template is heated to separate it into single strands. The next step is annealing. Now the temperature is lowered, allowing the primer to bind to its complementary sequence on the single-stranded template. During the extension step, the DNA polymerase starts adding nucleotides to the growing strand at the 3' end of primer. The extension continues until a DDNTP is incorporated. As the DNA polymerase synthesizes the new strand, it occasionally incorporates a DDNTP instead of a DNTP. This incorporation results in the termination of the strand at that position, generating fragments of varying lengths. The DNA fragments are passed through capillary gel electrophoresis, during which an electric field is applied, causing negatively charged DNA fragments to move through the gel. Smaller fragments move faster, while larger ones move more slowly. Since each nucleotide is labeled with a different dye, the laser excites the fluorescent dyes attached to the DDNTPs at the end of each DNA fragment. As the fragments pass through the gel, they are detected by their fluorescent labels. If one fragment ended with DDATP, it reads adenine. This is how the sequencing machine reads the sequence. The data generated is analyzed using software to produce a readable sequence. The order of colors detected corresponds to the sequence of nucleotides in the DNA template. I hope now you have understood the principle of Sanger sequencing. To know more about molecular techniques, do subscribe to our channel. Thank you for watching.